That's awesome. Yeah. Well, is that your actual background, Di, or is that a,
Yes, that's my office.
because it's
I
looking
got a
pretty
tree.
spring-like there. Yeah. Very nice.
For improvement, you need a tree inside your office in the wintertime.
That's really gorgeous.
Mhm.
All right. Well, it is 9:03 and looks like we have a pretty good collection, both in Johnson Tower and online. So let's go ahead and start. Let me introduce and express my gratitude to Dr. Simon Evans for talking with us today. I'm going to give a little bit of Dr. Evans' background, if you don't mind, Dr. Evans, just to show you how very broad is his expertise. And also, he's a Washington State person and he has worked at WSU. So we're welcoming him back in one sense. He got his BS from Western Washington University. He's worked in the Seattle biotech industry and then he got his PhD at Oregon State. So he kind of defected for a little bit, but he worked it in molecular and cellular biology applied to neurochemistry when he was there, and then went to University of Michigan for a postdoc and a faculty position to look at neurochemistry of stress disorders, and then focused on multiomics, looking at the gut microbiome and dietary patterns and how they influence bipolar disorder. And then back to Seattle to work at the Institute for Systems Biology and be a clinical professor at WCU Spokane. Switching gears a little bit, then he got his MBA at University of Illinois, my alma mater, and is now CEO of Phenome Health. And Phenome Health really just grabbed our attention. It's a nonprofit organization focused on personalized health and health innovation. And if you have not already seen it, you need to check out their recent article in Nature Reviews on the transition from genomics to phenomics in personalized population health. So I'm really excited to learn more about what you and your group are doing, and I'll turn it over to you, Dr. Evans.
That's great. Well, thank you for the invitation and for the introduction. One correction, I'm not CEO of Phenome Health. I'm the executive director, but Lee Hood is the CEO. He is really the visionary behind the organization. I'm just trying to implement the vision that if any of you have interacted with Lee in the past or know Lee, you know that he lives 5 to 10 years in the future, and the rest of us try to make it happen. It's challenging and a lot of fun at the same time. Um, I will share my screen. Let's see. Can you see that?
Yep, that looks good.
Okay, go in slideshow mode. So yeah, again, great to talk to this group. From my background, I know that Diane is a computer scientist. I'm not sure how many people in the audience are. I am not a computer scientist. So I hope to give you an overview of what Phenome Health is doing, why we're doing it, what we're currently working on, but I'm not really going to go into technical detail on the projects. However, if there is areas of interest and overlap and mutual benefit to move forward in future conversations, I will engage the right people to bring the technical stuff to the conversation. So yeah, implementing the vision of predictive, preventive, and personalized and participatory health, which is a term Lee coined about 10 years ago, P4 Health, to really express the need to create predictive models, but also to focus on prevention and personalization as, medicine is trying to move towards. But the fourth P, participatory, being an incredibly important component that is probably the most difficult to implement, where, people really have to be engaged in their own health trajectories and take responsibility and take authority over their own health trajectories. So I think that probably is consistent with your own research as well. So as Diane said, we're a nonprofit, 501c3 corporation based in Seattle. We incorporated in 2021, began operations in early 2022. So we're just nearing our 3-year mark, or just hit our 3-year mark, actually. We have a pretty great team on the leadership. Lee, as I'm assuming many of you know, if you don't, look him up. He's a pretty fascinating guy. He's on the short list for the Nobel Prize every year for his invention of the DNA sequencer, and has won every other prize there is to win in science besides the Nobel. So pretty incredible to work with. Diane gave you my background. I've really switched from the front lines of science to more the kind of organizational strategy role over the last 5+ years. Noah is fantastic. Data science lead. James is a background in engineering and molecular biology and really leads our engineering efforts. BJ, who you see has the same last name, is his brother and is a pretty fantastic computer scientist and really helps us build our computational infrastructure and expertise in cybersecurity. Tim, a practicing physician and really engaging kind of, I would say, biomedical entrepreneur. And Jennifer is really a clinical psychologist, so probably has a background more similar to some of the students in the room and leads our efforts there. We also have a pretty great board. Jeff Wilke was the former CEO of Amazon Worldwide. He has incredible insights and advice always to direct us. Maria Klawe, very well connected, now president of Math for America, was former president at Harvey Mudd College. Roger Perlmutter was #2 at Merck for a while. He's an ex-postdoc of Lee's, so that's the original connection. But all of these people bring a lot of credibility, expertise, and fantastic advice to what we're trying to accomplish. I like this quote from Confucius: "A healthy man wants 1,000 things, a sick man only wants one," because it really underlies or underscores the importance of health and the need that all of us have to try to seek at least the best health we can to take our lives forward. So just to kind of frame why we're in this field, really is to move from a reactive disease focus to a proactive health focus in the entire health space, whether it's within the healthcare industry or outside the healthcare industry. You know, the healthcare industry itself currently has a massive focus on diagnosis disease, right? Find and treat. And very little of the resources go into prevention and proactive health optimization. This is a report from Deloitte a few years ago that projected with an increased focus on wellness and upstream of diagnosis, actually trying to keep people healthy while they're healthy instead of waiting till they get sick, could actually save trillions of dollars over the next couple of decades. But this isn't really, they don't really see this as a savings, but more of a health dividend that would be reinvested or could be reinvested into the health and wellness market upstream of diagnosis. And so this kind of paradigm shift to move resources from, you know, post-diagnosis to pre-diagnosis. And we really want to accelerate that shift. That's kind of what it's all about. So this is actually our vision statement. We envision a world where everyone is empowered by their own data to improve wellness and prevent disease. Really getting data, usable data, actionable data in the hands of the health consumer. We don't like to use the word patient because patients, I don't consider myself a patient unless I'm actually in the hospital or I'm ill. But health consumers or research participants, sometimes you become a patient, but a patient isn't a place where we all live. So to get into it a little bit more, there are a lot of predictive modeling efforts ongoing now, leveraging, you know, the hundreds of thousands of whole genome sequences that have been created, the tens of millions, hundreds of millions of electronic health records. But we see this as really just scratching the surface. You know, you're born with your genome. Your genome definitely sets you up with various health risks or health benefits or tendencies, but it's really an indicator or a map of what might happen. And then electronic health records capture your interactions with the medical system, so they kind of tell you what did happen. But in between that is where we see phenomics. And there's a whole bunch of stuff that happens from the time you're born to the time you die. I think your own work in the behaviorome, fits very nicely into this, even though I don't have a spine of a book called The Behaviorome, but maybe I should. And I think, but elements of this, I think, capture that. So phenomics we see is really, shedding light on how, when, and why we move and from genetic risk or genetic predispositions to actual health outcomes, kind of the manifestation of genetic risk. And they also help us pinpoint what we can do about it to intervene and correct health trajectories. So we're measuring a lot of stuff. There's more stuff to measure, and it's expensive. So we're measuring as much as we can with the resources that we can secure. But this is kind of our standard current plate of things that we measure in our ongoing studies. A whole genome sequencing, obviously there's a lot of value in doing that. There's a lot of information in the genome, but it's limited, as I just said. Everybody understands that social determinants of health are important, so we capture as much of that as we can. We do, through partners, have access to electronic health records, so we integrate all of those into our modeling strategies. Self-tracking, a field that I think a lot of you are pretty familiar with digital health assessments. Wearables and environmental sensors to the degree that we can get the data, self-assessments. The microbiome, we've historically focused on the gut microbiome, but are trying to expand that to vaginal microbiome in some studies, oral microbiome in some studies. The microbiome is a fascinating field, still in its infancy. When I was at Michigan and got into the microbiome a little bit as an extension of the dietary work, and got into the gut-brain axis a little bit and how the microbiome actually influences behavior and brain chemistry and brain function, I think is incredibly fascinating. There's a long way to go in understanding that. We also capture elements of brain health using cognitive assessments, interactive tools. Brain HQ is one tool we use from a partner at Posit Science, who has a brain assessment and brain training program, but we also try to capture behavioral assessments, mood assessments as well. And then, of course, blood, which provides the bulk of the data from a gigabyte perspective and a resource cost perspective. All of the clinical chemistries, proteomics, metabolomics, transcriptomics, antibody screening, immune system function via purification of PBMCs, the white blood cells in the blood, and looking at those profiles. Just a lot of information. We really see the blood as a window into health because blood bathes all organs. A lot of organs release signals into the blood, which can be very indicative of their own health state. And you can't, at this point, it's hard to target every single organ, so blood is a very nice proxy into the health of a lot of systems. And really all of this to avoid McNamara's fallacy. And the current healthcare system, if you go to your cardiologist and you have high cholesterol and they put you on a statin, their outcome measure is to see if your cholesterol went down. But a lot of things that can happen when you take a statin. I mean, you could impact kidney function, you could impact risk for diabetes. If you go to a psychiatrist and they put you on an antidepressant and then the outcome measure is to see if your depression scores got better, there are other risks that can change that aren't monitored always by the system. So, trying to optimize for a single variable by doing these very limited interventions and very limited windows of outcome measures can compromise systems level optimization. And it's a very difficult problem. And we really try to approach it from that perspective. So just a word about, standard statistics. If you collect a bunch of data from somebody, standard statistics will assume independence of all those data points. But of course, we know that in biology, all these data points are connected through networks. So we try to take that into account, the connectivity and the interactions between any data in a biological system by mapping what we know onto that system. And we use knowledge graphs pretty extensively to do that. So the millions of data points that we collect get mapped onto knowledge graphs. We currently have the Spoke knowledge graph, which is a big one out of, I think, UCSD. It's a collaborative effort, an open source knowledge graph. We have that ingested into our core computational environment. We're currently figuring out a lot of mapping from a lot of the different omics and a lot of the different data streams onto the knowledge graphs. and then layering our own AI and ML analyses on top of these to try to extract previously unseen patterns or just higher resolution health insights. And this is, I think, something that probably interacts with or intersects with some of your own research. And I would love to have a further conversation with our technical people and your technical people on, kind of how we could be seeing this maybe through a different lens or incorporating some of the behavioral data that you have onto this as well. So really, we're just trying to extend the prediction models to maximize the value of the health insights we get, measuring all of this stuff. We have created our own core computational environment. We're still building it, but it's you're always going to be building your computational environment. It's never going to be done. And this is through a contract with another company that has built a lot of computational environments for companies like Amazon and Google, and sold those in-house. So we're really trying to build a sophisticated environment that allows us to ingest all of this multi-omic data, both biological, behavioral, environmental, perform the analyses that allow us to really predict health trajectories, but also inform decision points or intervention points where a health trajectory needs some correction. Looking at the manifestation of genetic risk, we have some papers on this where everybody has a genetic risk of something, but whether or not that manifests can be evaluated through capturing these type of data over time. Identifying actionable recommendations, as I've already said, and then really trying to identify both disease prevention and new therapeutic strategies to kind of take these forward. So this is all very high level. We have done some work in the past that we've already released. I think we've put out about 30 or so papers from past studies into pretty high impact journals. Really seeded by this initial project that preceded Phenome Health. This is a project that started at the Institute for Systems Biology, where Lee Hood and his partner Nathan Price at the time orchestrated a project to look at 108 individuals. They call it the Pioneer 100 because Pioneer 108 just doesn't sound good, so it's the Pioneer 100. And with 108 individuals that were monitored over a 9-month period, sampling blood every few months and doing proteomics, whole genome sequencing, looking at inflammation markers, looking at various behaviors. And that project was so successful that when it was over, the participants wanted to continue. So they actually spun out a company called Aravale, which some of you may have heard of. Aravale was a direct-to-consumer wellness company that lasted about 5 years. It was really ahead of its time and just couldn't get the price point down to a consumer-facing model, but offered services where they would do multi-omic analyses and behavioral coaching so that they actually would identify actionable results, actionable possibilities from all of the analyses, and then have behavioral coaches that would interact with the clients directly. And saw over the period of time of the existence of the program, biological age measures. Everybody has a biological age measure, but our own measures of biological age, which is the age kind of your body says you are, not your chronological age. If in women, those measures went down by 1.5 years for every year in the program, and in men, they went down by a year for every year in the program. So there was some success. There was decrease in risk factors and incidence. So it was a great program. But beyond just being a consumer program, it actually captured data on about 5,000 people over the period of time. So we now have that data set of 5,000 people with all of these multi-omics measures, many of them longitudinal, that we have been able to use to build out a lot of the pipelines and a lot of the tools that we're now using to take forward into future studies. A lot of wellness metrics came out of this. I already mentioned biological age, also biological BMI, which is and of a measure of metabolic health, similar to what standard BMI is trying to capture, but standard BMI misclassifies about 30% of people. And we identified a much better metabolic measure, not as cheap as weighing somebody and, and taking their height, but it has a lot more insight into the metabolic health. Some interesting cases of cancer diagnosis. So during the program, there were patients that progressed, or participants, I should say, that progressed to a cancer diagnosis. And when we looked back in time at their multi-omic data, pre-diagnosis, 3 months pre-diagnosis, you could already see pretty extensive disturbance of several networks. 5 months, 5 months, even close to a year, you could see some disturbance of some networks. And it took, going back more than a year before you couldn't detect these signals. So really making the case that high dimensional analysis, prospective analysis can really identify problems much earlier, which is no surprise, I think, to anybody. But it's good to see that actually manifest in the data. So, like I said, there are over 30 papers published. Here's just kind of a snapshot of some of them. This one up in the top left, the transition from genomics to phenomics and personalized population health, is what Diane referenced at the beginning of the introduction. The paper we published really summarizing all of this last year. Multi-omic signatures that correlate with Alzheimer's disease, some whole genome sequencing that actually allowed early detection of hemochromatosis, which was a very interesting paper to identify people that would never have known they were on the road to hemochromatosis had it not been for some of the multi-omic analyses. And that's a very easy disease to treat. It just requires donating blood every few months. So actually captured and prevented some chronic illness there. Interesting paper on the correlation of multi-omic data with genetic risk, kind of this idea of manifestation of risk, where you can see manifestation of genetic risk in the metabolome and in the proteome well in advance of any clinical symptomology or diagnosis. I already mentioned biological BMI. There was an interesting paper on genetic risk of LDL. So if you have a high genetic risk if you have a genetic risk that puts you at a high probability of high LDL, and you have high LDL, you probably need a statin to take it down. But if you have the same high LDL without the genetic risk for that, then a behavioral approach is more efficacious and you don't need the statins. So it provides another example of why you would treat people differently based on their genetics and based on their manifestations. Clinically, we see a clinical symptom, everybody gets treated the same if they have that symptom, but that's not the right way to do it, and we're trying to create the tools to do that better. Some interesting gut microbiome papers that both associate with healthy aging and actually predict statin response, and I'm sure this will be true for a lot of other medications as well, where the gut microbiome can provide insight and a window into the efficacy of given drugs. Just like pharmacogenomics is a big field where if you have given variants, then we know certain drugs aren't going to work for you and other drugs are going to be better, even though that's not widely deployed in the healthcare system. I'm not sure why, since it's been proven to be cost effective in pretty much every study that's been done. But similarly, the gut microbiome can be predictive of what drugs may or may not work, because the gut microbiome plays a role in digestion and metabolism of these molecules. So a lot of papers out there. If you're interested, I can send a list or you can find them, I'm sure. So what we are doing now in Phenome Health is really building the core assets and capabilities that we need to take this forward. The first being an education platform. So in order to engage participants in your studies, in order to identify physician champions who can push forward, these ideas, we need an education platform. Lee's wife, who actually passed away just recently, was a teacher all her life, and education has been important to Lee for a very long time. He established an education group inside ISB, and they have curriculum all over the world, actually, high school curriculum and college curriculum. And we are not yet big enough to really have an education platform built out, but it's important to us, and we are working towards building that out. The next asset is really establishing a biobank for all of our studies. So every time we meet with a partner or a prospective partner who's been doing, some kind of longitudinal work, and we ask them, have you been saving samples? Sometimes they have, sometimes they haven't, but there are a lot of great biobanks around the world. So we're not just building our own, but we're accessing, other longitudinal biobanks, which really give you the ability to do a 10, 15, 20-year longitudinal study in a year, because the samples have been collected and they're already there. All of this to generate a massive longitudinal data lake. Talked about the multi-omic data, the digital data, the clinical data, the self-reported data, which really are the major thing that goes in to help us model the health trajectories, and then building the computational architecture in a secure environment to get all that done, using that computational architecture and the data to drive the generation of the models, the health prediction, the recommendation engine, and then finally a commercialization platform to take it back to society. So Lee's MO has always been to develop technology to a point in a nonprofit environment or an academic environment, but then spin that out to serve society, because you can't serve society with a technology on a grant. You can't sustain a new technology without spinning it out and taking it back and allowing access by society. So that is kind of the loop of then feeding back to the health consumers in the first place. And we're currently building out all of these components, really focusing right now on the data lake and the computational architecture and the personalized models. But we've already spun out a couple of small things commercially to try to carry some of this forward, and are building out the educational platform as well. But there's a lot to do. We're not doing this alone. So we're working with a bunch of partners. And one in particular, we've created a strong partnership with the Buck Institute for Research on Healthy Aging in Novato in Northern California, just north of San Francisco. Lee and the CEO of the Buck, Eric Verden, are the directors of this institute called the Center for Human Health Span. And this is really acting as a platform for us to kickstart projects and move them forward. And I'll get into those a little bit. But the partner ecosystem to date, who we're currently working with, the Buck, obviously, who I just talked about, also a group called Guardian Research Network, who is a consortium of about 150 rural hospitals in the Southeast United States. They don't own the hospitals, but they have agreements with the hospitals to access all of their health records. So they have about 30 million patient lives that they have access to, and they provide a window to identify and recruit patients and participants into studies, because they have the EHRs. We can identify pretty quickly, and target populations to engage it and enroll. Also, Hudson Alpha Biotechnology Institute in Alabama has a small clinical research institute and a rather wide network throughout the Alabama region, who we engage for cohort access as well. So all of these three are really our major current players in identifying and engaging participants. And then, we generate our own phenomic data from that. But as I said before, also accessing third-party data from other biobanks and other data sets. UK Biobank has a ton of data now. All of us is getting up to speed. Finnish Biobank is another organization we're collaborating with to access historical sets of samples relevant to both Alzheimer's disease and diabetes, and taking some of our projects forward. We're working with Technicity to build our platform, who's the company I referenced earlier, who has built quite a few platforms and commercialized them with big players. We're working with other organizations on the predictive modeling, pattern computing being one. Also talking to a few others that we're moving agreements forward with. The buck is nice because it has experimental systems. So we're a computational biology organization. We don't do wet. We contract all the wet lab out. The buck has about 300 people, 24 faculty, and I'd say 22 of them are wet lab scientists. So they have cell models and mouse models and worm models and fly models and all kinds of preclinical modeling where we can take hypotheses generated from the computational biology that we do and actually take them back into the lab and close the loop and enter the preclinical systems to further refine those hypotheses and identify targets. And then for the return of results platforms, actually taking things back to society, we're working with a couple organizations. Brain HQ is a cognitive health focused organization, Posit Science, and Phenome AI is actually one of our own spinouts in kind of the AI med-splaining space, working to really empower physicians to make every physician a domain expert in every field, because AI can help facilitate that. And then intervention platforms. Posit Science also has a cognitive intervention. P4 Bios is a new peptide drug company that has a very new and different model to be able to shift cells from one state to another. So really trying to identify health states that need to be shifted to and current states that they're in, and identifying the right targets and cocktails to make that happen. We are talking to some health systems. We don't have any agreements in place, so I'm not naming any of them, but we're talking to several, also concierge medicine type physicians and clinics, and obviously health consumers, all supported again by an education platform. And so Hudson Alpha actually has a great education platform in Alabama. ISB has a great education platform. Buck has an education platform really tailored more towards graduate students and scientists than the general public, but I'm trying to bring together the right players to generate the appropriate materials and education. So that's kind of a snapshot of what we're trying to do. Now I'll just spend a few slides on some ongoing projects. Again, really at the overview level. I'm not going to go into any deep detail. The first is a type 2 diabetes project, which we've been awarded recently. Cash flow has not started yet. I anticipate it will be in the next month or two, but this is a $21 million award to us and Guardian Research Network, almost evenly split, not because it's evenly split, but because Guardian Research needs the funds to identify and recruit and engage all the participants and bring them in to the clinical environment and consent them and all of that. And then once data starts to flow and blood comes out of the arm, then we kind of take over and deal with all of the sample processing logistics and data flow logistics and data analysis downstream of that. So the plan is to recruit 1,000 type 2 diabetics and a couple hundred non-diabetic controls. Originally, it was going to be a longitudinal study, but through constraints of the award or awarder, we are really doing a cross-sectional, high-dimensional multi-omic analysis of diabetes for the purpose of identifying molecular signatures of subtypes. So there's a growing literature in type 2 diabetes that there are probably 5 or 6 subtypes, all with unique clinical trajectories, and those subtypes are currently defined by a handful of clinical markers and clinical outcomes. They're not really defined via the underlying molecular signatures that would actually provide insight into the mechanisms of those subtypes. So the purpose of this project is to identify those molecular patterns and molecular fingerprints underlying each subtype. And that will actually feed into the next project that I'm going to talk about here, which is an award from ARPA-H of up to 52 million over 4 years, mostly in collaboration with the Buck, but also with a couple other organizations. This is a 5-project, very complicated award, and I have one slide on each project to go into. just to kind of give you a flavor for what they are. Project one is to really understand, and I know, Diane, you probably appreciate this because I saw it in some of your own papers, referencing like Nyquist rates and things, that we really do not understand the underlying signal structure of a lot of the multi-omic data that we're measuring. So we are going to do about 125 person high frequency trial or observational study where we bring people in and we're doing blood sampling 5 times a day across 4 days. So pretty intensive for them. It's not minute by minute or hour by hour, but a few hours between each sample, to try to map out the underlying signal structure of all of the blood-derived multi-omic signals we're deriving. We're doing continuous monitoring with wearables and other digital health as well. But for the the biological measures, getting as tight as we can without bleeding people to death and staying within IRB constraints and ethical guidelines. In addition to the 4 high frequency days, there will be sampling once a week for 10 weeks. And during 2 of those weekly visits, we'll run a perturbation. One will be a VO2 max test, so an exercise perturbation, to look at response of the multi-omic system under that kind of stress load. And the second being an oral glucose tolerance test. These are not diabetics, they're healthy people, but we'll run an oral glucose tolerance test to give a glucose load and look at the system response and recovery to that stress as well. And so really trying to map out in a pretty sophisticated way the underlying signal structure as much as to the degree that we can with all of the omics that we're looking at in all these other studies. The second project is just a computational, I say just, it's a completely computational effort to transform a lot of the existing data silos around the world into more connected and interoperable resources. This is led by Noah Rappaport in our group, who's a fantastic data scientist and expert machine learning. And to take data sets like the Airveil data that I talked about earlier, there's a 10,000 person data set in Israel that has done a lot of phenomic assays, but not so much longitudinal. There's, the UK Biobank data, there's Twins UK data, there's the Olivus data, there's tons of data out there. It's very difficult to leverage it all in to the highest value that we should be able to do because of the interoperability. And so trying to create machine learning models and portable models that can actually allow ingestion of all of these data and gain more insights from them. The third project is actually leveraging the type 2 diabetes project I talked about earlier. So this is a project working with a biobank out of Canada called the PROMISE study. They have a biobank about close to a 20-year, I think an 18-year biobank now, where everybody was recruited with risk for type 2 diabetes, but not yet diagnosed with type 2 diabetes. And that was validated at their enrollment point, running an oral glucose tolerance test and showing a euglycemic response to the test, which is kind of a gold standard for type 2 diabetes. And they have brought people in every 3 years, have saved the samples, including an oral glucose tolerance test at each of at least the pre-diagnostic time points once they have a type 2 diabetes diagnosis. OGTTs are contraindicated, and they didn't do that if they came in with a type 2 diabetes diagnosis. But we are accessing this data to run our multiomics on as much as we can of all of the people that have transitioned to a type 2 diabetes diagnosis and an equal number of controls that have not. And then we're using the post diagnostic data or samples and data points to map on the molecular subtypes that we get from our other diabetes projects. So we'll have these molecular fingerprints and molecular subtypes of type 2 diabetes. We're going to map that on to all of the participants that have converted to type 2 diabetes, and then look back in time to try to identify subtype specific pre-diagnostic biomarkers or signatures that would inform future health trajectories to try to better identify appropriate intervention, subtype specific intervention points. And the nice thing about this study is they don't just have all the baseline samples, fasted samples of people before diagnosis, but they actually have the OGTT response, because sometimes you need to stress a system in order to see what's wrong, right? I mean, if you have a chair with a loose leg, it looks fine, it's in the corner, but you go sit on it and put it under stress and it might collapse. So, I had a mentor at Michigan who liked to say, in order to understand a river, you have to watch it run, which is true. You can't just, sometimes you just don't see the signal you want in a homeostatic state. You need to perturb the system. So this provides the mechanism to look back in time and do that, which is fantastic. Our fourth study, just briefly, is a very high risk study, but trying to leverage wearable technologies, combinations of CGMs, continuous glucose monitors, and wrist or finger wearables in type 1 and type 2 diabetics, in combination with gold standard tests of pancreatic beta cell function, and try to map out the digital signatures of pancreatic beta cell function, which may or may not exist. But it would be fantastic to have a non-invasive, scalable, distributable assay, digital-based, to try to monitor beta cell function or other diabetic manifestation risk factors in those at risk, so they wouldn't have to go in and get an OGTT or get a blood draw in an intensive invasive test to track the progression of the disease towards a diagnosis. So that's kind of the goal of that project, probably the most high risk one. And then finally, it all really comes together in the fifth project, which is led by Nathan Price at the Buck, who used to be at ISB, of building an N of 1 analyzer, really meaning to be building an engine that can ingest multi-omic data at various levels of missingness or completeness, and actually output accurate health trajectories and informed intervention points, which kind of underlies everything we're really trying to do. And they have a strategy to build this. So, we've obviously built models to date, but models can always get better. The famous George Box statistician quote that all models are wrong, but some of them are useful. So we're just trying to, improve our models as much as we can over time. Finally, we have a pilot study funded. We don't have the full study funded for an Alzheimer's disease with our partners at Hudson Alpha in Alabama, to do a very similar type of program that we're doing with the type 2 diabetics, screening patients at risk for AD pre-diagnosis, and trying to identify better pre-diagnostic markers, but not just markers, but actually, intervention by doing the full multi-omic analyses, really identifying the mechanisms of progression and identifying intervention points. So that is about to start. We're, I think, recruiting the first patient for that in April. This is the last slide. All of this really to transition from what health looked like yesterday to what we want it to look like tomorrow. I mean, it's always been disease-centered, and we're trying to move towards a wellness and prevention-oriented system. It's always been episodic. Maybe you go to the doctor once a year and get your physical, and they might talk to you for 5 minutes, to more of continuous monitoring, self-empowered continuous monitoring. It's always been very centralized. We're trying to move towards a distributed model. Everything's in the past is based on population averages. We're trying to move towards personalized approaches, trying to move from single provider to really crowdsourced systems where leveraging AI and new technologies to increase the domain expertise of any individual, and moving away from the fee-for-service model to a real value-based system. So that is the overview that I have. I know I probably only scratched the surface on some things, but I hope there's enough time for some discussion.
Thank you so much, Simon. I filled pages with insights from your comments and ideas from the directions where you're going, and quotes. I like the one from your mentor. Let me open it up first to individuals on Zoom. I guess you can unmute yourself and ask a question or raise your hand while they're figuring out the technology and the question. I was wondering, you mentioned that you are integrating wearables, continuous monitoring with wearables. I was wondering if you could expand, since that's the area we're looking at, what types of data markers you're collecting with wearables and how they're being integrated.
Yeah, so James Yurkovich, our lead of the engineering group, is evaluating a lot of wearables, Whoops and Oura Rings and Fitbits and Garmin's and Apple Watches. And actually, we have a collaborator, Stephen Friend, who used to be at Apple. He has a lot of insight into the raw data that comes off of these. We're trying to get as close to the raw data as we can, not just use the commercial insights and outputs. We're
Yeah.
really interested in sleep quality, right? I mean, that's such a huge factor in health. And it's, I mean, this is your field probably more than mine, but Oura Rings might be better for that just from a compliance perspective. A lot of people don't like to wear watches to bed. So we're trying to get as much of the raw data and generate our own algorithms, as well as use the algorithms that these companies have already created. But also, yeah, CGMs for continuous glucose monitoring. They only last a couple of weeks, as you know. We'll be slapping those onto people a few times during some of these studies. Uh, we talked to this company recently that has an eyeglass technology, which actually is pretty cool because it's an attempt to passively monitor diet behavior. So it has a sensor to monitor the chewing muscle, right, in your jaw, and then triggers a image capture from the glasses to capture both the timestamp of when you're eating, how long it's taking you to eat, how much you're chewing,
Mhm.
and then the image analysis of food still isn't perfect, but it's better than nothing. So there's that. But the other thing that the glasses can measure, which I'm really interested in, there's this whole field of study on facial muscles being activated under different mood states, right? I mean, you can tell if you're kind of angry or happy or sad or depressed or whatever. And the glasses they're inward, the sensors are inward facing. So they capture contraction of like 35 different muscles or something and try to get an objective measure of mood over time, which I think is really cool. So that's another potential continuous one that we're trying to figure out how to build in, at least at a small scale in the beginning. So yeah, all kinds of technologies out there that are emerging that are really cool.
What a wonderful sandbox you play in. I mean, the tools are are just so creative. But I see, Larry, that you are on video. Do you have a question?
Raise my hand. I guess some of my former research is in graph analytics, and I noticed you're using the knowledge graph. And I think most people are using that today to provide context into, like large language models. But I was wondering if you had any plans for actually doing analysis of the graph itself and looking for patterns that might be correlated with the various health outcomes and interventions that you're working on.
Yeah, absolutely. Again, I'm going to default that I'm not a computer scientist, and I'm not going to be able to go as deep on that question as you want. But yes, we are doing that and actually using some kind of image analysis techniques to evaluate the patterns in the graph. And we are using I think Spoke is typically used with Neo4j. We're actually using ArangoDB, which allows you to store a lot of information on the edges, not just in the nodes. So we're adding a lot of value or a lot of information to the graphs through that strategy. And yeah, if you want a detailed discussion on that, I can put you in contact with the right person. But yes, we are considering that.
Okay. I guess one other question, if I can, in terms of the wearables, the data, I guess two-part question. So one is what kind of wearables are you using? Is a smartwatch enough, or do you need more dedicated, uh, you know, wearables, on wrists, waist, ankle, that kind of thing?
Yeah, we haven't gone to the waist or ankles, wrists and fingers, mostly through the Oura Ring or Oura Ring-like technologies and smartwatches primarily, and then the CGMs are the primary. Those are the wearables that are really put on everybody. We're exploring every technology that we run across that looks informative at the scale that we can.
And last question, is any of your data available yet?
It will, so the Airveil data is partially available. Some of that's in the public domain from previous publications. We licensed that from Airveil. We do not have the right to, it's not our data. We don't have the right to just give it away. But I think at least 1,000 of the 5,000 complete data sets, the most complete data sets are publicly available. We do have plans to make a lot of the data from the ARPA-H award available. We're just getting ready for first patient in on that study in April. So our first patient in for the longitudinal diabetes study will probably be in like July. For the ARPA-H study will be in April. For the Alzheimer's study will be in April. So we're really building the infrastructure and gearing up to generate the multi-omic data. It's not there yet. Outside the Airveil data, it's not there yet. But it will be available.
All right, great. Thank you.
We're a research nonprofit, so.
Sure.
I have another question, then I'll turn it over to Johnson Tower Group. I like that you're going to be integrating large multi-omic data sets and, looking at techniques for imputing missing data features, especially given the diversity across the data sets, seems very reasonable. One thing I have found when we try to build models across even multiple cohorts of individuals collected internally, so same experimenters, same people designing the apps, is that our machine learning models do not always generalize well across cohorts. They generalize worse than from one person to another within the same cohort, maybe due to just slight differences in the collection mechanism or the participant base, or even the one generation of Apple Watch, say, to another. And I'm wondering, in your integration of these large data sets, if you've already looked at that, the generalizability of some of these findings and models across different data sets.
Yeah, I wish Noah was here to answer that question. I can't, I just don't know. I know that Noah and her partner in crime, John Earls, at the Buck, John was actually responsible for building pretty much all of the Airveil computational infrastructure and is now working on this project. Are incredibly talented people, and I can only assume that they're doing the right thing, but I can't answer your question directly.
We will look for papers to come on that because integrating them is so important. There's so many dispersed data sets out there that we need to harness, and that harnessing them in a cohesive way is really hard.
Yeah,
So
okay. I mean, I would love, if you want, I mean, I would love to have follow-up conversations and actually bring in Noah and James to address the more technical.
I would very much enjoy that because there's so much data out there that we need to bring together, but it has to be done in a careful way because of those slight differences can really cripple you.
Yeah.
But Johnson Tower crew, do you have some questions?
Okay, go ahead. You go first.
You'll have to speak up very loudly, I think.
Hi. Great presentation. It was really, really interesting. I had a question about the wearables. So with the Oura Ring, is it using, and I'm less familiar with that, but is it using movement as a proxy for sleep the same way in the actigraphy does? Or is does it have a different method of measurement in that way?
Yeah, I mean, it has movement sensors, it has temperature sensors, it has the heart rate sensor. So it uses all of that information. I don't know the Aura proprietary algorithm, but it integrates data from, and it's not just movement. It's movement plus temperature plus heart rate.
Thank you.
Simon, thank you so much. That was, I don't know, it's just like so incredible to see all that information that you're bringing together. It's really exciting. I can't wait to see where you are in another 5 years. Well, hopefully sooner. But I guess my question is, in terms of, I know like you're doing a lot of oversampling right now, but in terms of like the big picture, if you're going to roll this out to the population, how much data do you see needing to get on each person? Like, how often are you going to have to get all types of data on everyone, or like what is your envision there?
Yeah, I mean, the whole strategy is to identify a data reduction or dimensionality reduction strategy, right? To identify what are the most informative features, which is probably going to vary across person and across health states. But we don't anticipate collecting all the data on everybody forever, no. The other component to that is not just, identifying the features that are most informative, but the adoption of new technologies that are constantly coming out that are moving more and more towards, home-based distributed technologies. I mean, even at home now, what people have smart scales and blood pressure cuffs and SpO2 sensors that, I mean, those didn't exist in the home, 10 years ago. So I see technologies all the time. I saw one yesterday that was this really cool microsampling device. There's a lot of microsampling devices now that, are actually taking interstitial fluid and can do onboard assessment of 1 to 16 molecules at a time using microfluidics, using very scalable, high throughput manufacturing techniques. So all of that kind of coming together, not just identifying what are the most important features, but driving the technologies forward to do, more accessible, cheap sampling.
Hi, amazing presentation. I think it's so cool to see the integration of everything. With the integration and the future rollout into commercialization, I was wondering if you had any idea what the feedback to the consumers would look like, whether it would be through an app or through some form of an integrated report, and if they would need a medical professional to go through feedback with them, or how you plan on making it understandable for the consumers.
Yeah, so that's a really good question. So Aravail actually gave information directly back to the consumer. It was not medical information, it was health information, right? If it's information that actually influences a medical decision, it's under FDA control and it has to go through a doctor. And Aravail did not do that, and that was actually one of their shortcomings. So we plan to do both. We plan to build return of results platforms, both for the health consumer that can go directly back to them, boiled down in very informative, actionable ways, and through the doctor, which may flag various risk factors or various treatment strategies for them to consider in their practice. Apps, probably an app right now. We already have we have a chat bot that has been trained on a lot of the medical literature already that you can ask questions of, and it may, text-based kind of chat bots seem to be gaining momentum and may actually be an easier way to deliver information than via an app. Everybody's got an app. So unclear at this point, but we're pursuing both channels.
Okay, I think we're good here.
Okay,
you.
one more question from me, and then we'll probably close it because I know people have class schedules. I was wondering if in your future work, you're going to be looking at multi-generational participants. I know that here at WSU, there's a lot of people with expertise in epigenetics, and Larry has looked at that too. And since you're connecting, environment and behavior and health, it would be interesting to see that full cycle. Is that something that is likely to be considered?
Yeah, it's not baked into any plans to date, but Lee has brought that up many times, the power of, family analyses and multi-generation analyses. He did a big bipolar and a big Alzheimer's study, I don't know, maybe 15, 20 years ago, using families and that strategy, not methylomics or not, I think it was just genomics, but we do have plans to look at methylation patterns and the methylome as well in at least the time study, the Project One from the ARPA-H study, and probably the diabetes and Alzheimer's studies as well. We don't have it built into our recruitment strategies for multi-generational, but it is definitely something on the radar.
Di, did you have a question? Oh, okay. Well, thank you again for your time. I would love to talk more. As I mentioned, we even have a mechanism to support some of your researchers who are interested in connecting some of the genetics and behavioral piece that we can talk more about. And just fascinating work. Thank you so much.
I guess what I didn't say relative to that is we have taken interns from a master's in health program in Canada. And, if you have students, graduate students or junior faculty or whatever that would want to kind of engage directly, that you would support through your program rather than supporting someone on our team, supporting someone on your team and embedding them with us, that's another possible avenue, which would be great to discuss.
That is great. We will talk about that. So thank you. We'll follow up.
Okay.
Okay. Thank you again. Everybody
Okay.
enjoy the rest of your day.
Thanks, everybody. Bye-bye.
Yeah.